Namaskar, welcome to PK's classes. Today we are going to discuss about the mechanisms of anti-cancer drug resistance. There can be at least 7 mechanisms of anti-cancer drug resistance. There can be increased efflux. Let us see the diagram. There can be increased efflux due to overexpression of the drug efflux pumps. There can be mutations in the drug target, alterations in the drug target. There can be loss of receptors. Number 3, there can be altered apoptotic pathway. As we know, most of the anti-cancer drugs are cytotoxic, which induce apoptosis. And if the apoptotic pathway is altered, then that will lead to resistance. And apoptosis occurs due to inhibition of DNA synthesis or DNA damage. So, if there will be enhanced DNA repair in response to the DNA damage, that will also lead to resistance. There can be decreased drug uptake or penetration into the cell, and there can be altered drug metabolism. In addition to this, there can be another cause of drug resistance, the cytokines and growth factors. If their level will be increased, then that will also lead to cancer drug resistance. So let us discuss them one by one. First, there is increase in efflux. So this efflux is done by a superfamily of ATP binding cassets, ABC proteins, or P-glycoprotein, PGP. So when there is expression of ATP dependent multidrug transporters or membrane proteins encoded by multidrug resistance 1 gene, they will cause the efflux of the drug from inside to outside the cancer cell. So that causes the resistance. Because of extrusion of the anti-cancer drug, the intracellular drug concentration becomes very less. It goes below a cell killing threshold. That leads to resistance. So drugs which are extruded by P-glycoprotein are the anti-cancer antibiotics like doxorubicin, daunorubicin, actinomycin D, the plant products, the plant-derived anti-cancer drugs like vinblastine, vincristin, paclitaxel, tenofovir, etoposide. The second mechanism of resistance is altering the drug target. So the target can be a receptor, can be enzyme. So under this we will discuss in 3 headings. First one, if there is increase in the drug target level. Suppose there is increase in transcription of the gene that encode the target or the enzyme, there is gene amplification. So that leads to increased levels of the target. And since the drug concentration is limited and the target level is increased, so the drug will not be able to block the additional targets or enzymes synthesized, and that leads to resistance because the cell will effectively overcome the inhibitory effect. So example, methotrexate, which is a folate antagonist. So if there will be more folate synthesis, then methotrexate resistance occurs. Second mechanism under the altering the target is decrease in the target level or loss of the drug target. Sometimes there is reduction or loss of the target or receptor so that the receptor is no longer available to be blocked by the drug, and that also causes the resistance. And the most common example is in breast cancer. Tamoxifen, the anti-estrogen drug, resistance occurs. When there is loss of estrogen receptors in the resistant tumor cells or the breast cancer cells, patient no longer respond to tamoxifen because there is loss of estrogen receptors. That leads to tamoxifen resistance. The third mechanism under altering the target is mutation of the gene encoding the target. Mutation of the gene. So if there will be mutation of the gene encoding the target, then the target may disappear from cancer cells and cells become insensitive to drug, resistance occurs. For example, mutation of topoisomerase 2. Topoisomerase 2 is inhibited by the anticancer, cytotoxic anticancer drug plant derivative like etoposide. So mutation of topoisomerase 2 leads to its disappearance from the nucleus, and cells, the cancer cells, become insensitive to etoposide, and that results in etoposide resistance. Now we'll come to the third mechanism of anti-cancer drug resistance, that is defects in the apoptotic pathway. Most of the anti-cancer drugs, they cause DNA damage, and that leads to activation of the negative regulator of cell cycle, that is p53. So there will be p53 mediated G1 cell cycle arrest, and that leads to apoptosis or cell death. So there will be prevention from production of the cancer cells or the defective cells. But when there is mutation in p53, so the p53 function is lost, so there will be no negative regulation of cell cycle, so it will allow cells even with damaged DNA to continue replicating. So that causes the resistance to drugs which act by damaging the DNA and therefore inducing apoptosis. So for example, reduced expression of p53 in human breast cancer cells causes resistance to paclitaxel, 5-fluorouracil. So under this, p53 is very common one. Other genes involved in resistance can be HRAS, BCL2, BAX, etc. Now the fourth mechanism of anti-cancer drug resistance is enhanced DNA repair. As already we have discussed in last point, that when the anti-cancer drug causes DNA damage, that induces apoptosis. But if in response to DNA damage there will be enhanced DNA repair, that means there will be resistance to the anti-cancer drug. So we know cisplatin, a cytotoxic platinum compound, where the platinum forms adducts with DNA, so there will be DNA lesions which induce apoptosis. So resistance occurs when there is increased expression of xeroderma pigmentosum group E binding factor and excision repair cross complementing protein, which is a DNA repair protein. So they will, and XPEBF is a nuclear protein, and they recognize the cisplatin induced DNA damage, remove the cisplatin DNA adducts, repair the cisplatin induced lesions. So cisplatin induced cytotoxicity is inhibited, and that leads to resistance to cisplatin. Now we'll discuss the fifth mechanism of anti-cancer drug resistance. The fifth mechanism is decrease in penetration of drugs, and it depends on the size of tumor, because chemotherapy is more effective for smaller tumors. The central portion of large tumors tend to have a poor blood supply, and drugs will have limited access to the central area of a tumor, a large tumor. Therefore, chemotherapy is more effective for smaller tumors and becomes less effective for large tumors, and that may be a cause of failure of drug action, that is resistance. Similarly, for brain tumors, the drug must act centrally after crossing the blood-brain barrier. So if the drug cannot cross the blood-brain barrier, then it will be ineffective against the brain tumors, and that can be a cause of resistance. And decrease in penetration of drugs or drug transport can also be caused by genetic variations in the transporter. So if because of the genetic variations in a certain population, the transporter, say, is less available, then that may decrease the intestinal transcellular permeability, so that will decrease the drug absorption and biodistribution to tissues, and that can cause resistance. Now the sixth mechanism of resistance, altering metabolism. So again we will discuss it under three heads. One, overexpression of drug metabolizing enzymes. So that will, if there will be overexpression of the enzymes metabolizing the drug, then there will be increased metabolism, so increased elimination, decrease in drug concentration, so that will lead to resistance. For example, increased expression of dihydrodiol dehydrogenase in ovarian carcinoma cells increase the metabolism of cisplatin, so that causes the resistance to cisplatin. The resistance can also occur due to underexpression of drug metabolizing enzymes in case of prodrugs. So inactive prodrugs which are metabolized to become active, in those cases underexpression of drug metabolizing enzyme can lead to resistance. For example, cytarabine is a prodrug which is converted to cytarabine triphosphate by deoxycytidine kinase. So initially it is converted to monophosphate, then diphosphate, then triphosphate. So if there will be underexpression of this enzyme, cytarabine cannot be activated to cytarabine triphosphate, so that leads to cytarabine resistance. The third mechanism under this altering metabolism can be genetic variations in metabolic enzymes. So there can be either genetic variations in drug transporter, which alters the or affects the penetration of drugs, and there can also be genetic variations in metabolic enzymes. So mostly the enzymes affected are the cytochrome P450 group of enzymes, whose structure and expression are genetically differences shown there in the CYP450 group of enzymes, and the example here is CYP3A4. So the drugs metabolized by CYP3A4, mostly the drugs which have shown affinity for P glycoprotein, they also show affinity for this CYP3A4. So there will be altered metabolism, and there can be altered clearance. There can be biliary disposition in hepatocytes, increased biliary disposition, increased urinary elimination, so the drug concentration will decrease, and that leads to resistance. The seventh mechanism of anti-cancer drug resistance, there can be increased expression of cytokines like interleukin 6 and growth factors like fibroblast growth factor. These cytokines and growth factors are the mediators of uncontrolled proliferation. So when they are expressed more, that can also lead to resistance. For example, in breast cancer cells, autocrine production of interleukin 6 activates the CCAAT enhancer binding protein family of transcription factors, which induce the MDR1 gene expression and causes resistance. Similarly, in solid and metastatic tumors, there is elevated levels of acidic and basic fibroblast growth factors, and that can lead to increase in tenfold resistance, tenfold increase in resistance occurs. And the example of drugs like paclitaxel, doxorubicin, 5-fluorouracil, there can be tenfold increase in resistance due to elevated levels of fibroblast growth factors. So suramine, a known inhibitor of fibroblast growth factor, can reverse this resistance. So this is all about the mechanism of resistance to anti-cancer drugs. Thank you.